Okay, so how are people doing with this question? You need more time? Huh? Okay, so Caroline. Who's Caroline? Corey. Oh dear. Okay, we're gonna try one more. Brittany. Ah, there she is. Okay.
I said it would hinder vesicular transport because it prevents the addition to like microtubules, and it increases the likelihood of catastrophe.
Perfect. Perfect. So the first thing to think about, so what Brittany said was it would inhibit vesicular transport because Staphin prevents addition of subunits to polymerized microtubules. And so if you have no microtubules, you can't have vesicular transport because that happens on microtubules. Did anyone have a different way that they thought about this? That's perfect. That's really a perfect answer. And so this is how I want you guys to be thinking when you're studying. Don't just think Staphin binds to tubulin subunits, because that's not going to help you to answer a question. I want you to think about what are the implications for microtubule dynamics. What do microtubules do? What are the effects on these processes? That's how far I want you to be going when you're studying. It's funny up here. I always hear conversations in the hallway. Okay. So far, this is what you need to know for next week. All the different, so there's two major panels in the textbook. And I've been telling people who've been coming to office hours, make sure you know those two panels. So like panel 1, that's all the concepts of the cytoskeletal dynamics, from nucleation to critical concentration. It's all covered in like this 2-page panel. And then there's another panel that has all the accessory proteins. And those are two very good places to start when you're trying to study and organize your thoughts. So really make an effort to look at those. If you don't have them, you can come to my office and take a picture of them, whatever you want to do. But they're useful. So we've got our key players, our major concepts. We talked about toxins, how they work. And then you should be thinking about these toxins and how they might be similar to some of the accessory proteins we spoke about, right? We spoke about nucleating proteins, proteins that bind to subunits, severing proteins, side-binding proteins, end-binding proteins. And then we were starting to talk about proteins that organize cytoskeletal filaments, and then everyone looked brain dead on Tuesday. I've been feeling brain dead. It's that time of the semester, huh? Who's done? I'm done. Thanksgiving next, like 2 weeks, right? Let's just go with it tomorrow. I'm going to do Thanksgiving tomorrow. If anyone wants turkey. Actually, I don't think we do vegetarian Thanksgiving. Anyways. I know, vegetarian Thanksgiving is weird. We don't even do toast turkey. We just do lots of baked vegetable deliciousness. Why? Why do I do vegetarian Thanksgiving? It's a long story. I used to be a vegetarian. So here's the story. I used to be vegetarian. I started being vegetarian at 14. And I stayed vegetarian until I was pregnant with my 3rd child. And then it was a boy, and I think boys must require more meat. So I don't know what it was, but I suddenly was craving live animals. Like, so I started eating meat again then. So that was from 14 till 32 or 33. And but before that happened, we had neighbors who were strict vegetarians. We started doing Thanksgiving together because all of our families were far away. And it turned into this like 40-person huge Thanksgiving party. And so it just became like the vegetarian Thanksgiving. Now we've got kids, everyone's got kids and families, so people do their own thing, but these neighbors still come along with my parents, which is interesting. Anyhow, my parents like turkey, so my dad's always like, is there going to be real food? I'm like, well, the other people will come if there is turkey, so we respect the other people. Hardcore. That's the story. Okay. So tomorrow I'll bring in turkey for everyone? No, I won't. Okay. I'm just totally like, end of semester punchiness. Yeah. Okay. So we talked about all these accessory proteins. So the things that we need to think about though is there's another class of accessory proteins. And they function in organizing these cytoskeletal filaments. Because if you just have microtubules floating in the cell, how are they useful, right? If they're not attached to anything, right? And actually, you asked about why organelles move a while ago. And as I was prepping for this lecture, I started thinking about that question again. And don't think so much about organelles moving, think about them being where they should be. Okay? And how if you have an organelle just floating in the cytoplasm, it's like, how is it going to be organized, right? Your ER is here, which is near your nucleus, and your Golgi is not too far away from that. Whereas if you don't have things kind of anchored to where they need to be, then they're not where they.
Would they also work to keep them then?
Exactly. So you actually have motor proteins holding onto organelles and the cytoskeleton to help keep them where they should be. And so we'll show some things hopefully today about that. Anyways, this is what happens sometimes. You ask me questions, and if I answer them inefficiently, then I keep thinking. So this is just showing how microtubules are anchored to the centrosome, right? These gamma-tubulin ring complexes, the nucleating proteins, are at the centrosome, and that is why you see your microtubules growing out of your centrosome. That is why your mitotic spindle is kind of anchored to duplicated centrosome, right? So let's just go back to intermediate filaments for just a minute. They also need to be somehow anchored to the cell, right? And so there are some accessory proteins that hold intermediate filaments together and also anchor them to microtubules and actin and kind of bring everything together in terms of the cytoskeleton. So here you could see in this picture, the pseudo-colored microtubules are red, intermediate filaments are blue, and then there's this plectin, which is green, and it's actually a cross-linking protein that helps to cross-link intermediate filaments with each other or bundle them with microtubules, actin, myosin, and the plasma membrane, right? Because if you don't have intermediate filaments attached somehow to the cell, then how are they going to play a role in stabilizing the structure of the cell, right? Does that make sense? Yeah. Good. So active filaments are also organized, right? So they have to be somehow interacting with the cell cortex, right? So they're organized into either bundles or web-like, gel-like networks. And so this is where we said, okay, remember you're learning about two different types of actin nucleating proteins. They're not interchangeable. They do different things. They function in different cellular processes. So formins make bundles, such as contractile bundles. And our complex makes webs or gels. And these different structures of actin function in different processes. So for example, Your cell cortex is when it's moving forward and you have a leading edge, that is nucleated by ARP. And that kind of actin structure is associated with motility. Whereas contractile bundles, sometimes you'll find them during movement, for example. You'll find them in the rear of the cell to help kind of scooch the cell forward. And that's made by hormone. So the nucleating proteins make actin filaments and certain structures, but not only that, you need these cross-linking proteins to bundle them and keep them in the proper structures, right? And so these are bundling proteins and gel-forming proteins. They're cross-linking proteins. Cross-linking, that means they bind to two things and stabilize their structure. So if you look here, this is showing 4 different cross-linking bundling proteins. They just play a role in organizing the structure of the actin. The actin binding sites are in red. And then the green are basically linkers, linking the two actin sites. So you can imagine one site's going to bind to actin here, and the other site's going to bind to a different actin. So, for example, spectrin, this is a very long linker, so this would probably bundle actin very far apart. Filament has this shape, and so it would probably function, and you'll see some pictures, in cross-linking something that's going to be like this, such as an ARP-formed actin gel. Fibrin and actin have small or no linkers, so they're going to bundle actin close together. So you have to visualize in your head, if the red part's actin binding, how actin would be spaced with each of these different proteins. And they all serve a different function. So what am I talking about? I don't know. We'll talk more about it. Don't worry, I'm not going to leave you hanging there. So bundling proteins, they cross-link actin filaments into a parallel array. They also determine what proteins interact with the actin filament and can therefore determine function. So different structures of actin are associated with different functions. And those structures are formed by these different bundling proteins. So based on that, you should be able to predict exactly, not exactly, but at least eliminate certain functions when you see a certain bundling protein. For example, myosin 2, a motor protein that interacts with contractile bundles, cannot interact with the tight packing of actin filaments held together by fibrin. Fibrin's tiny. Let's look here again. Where's fibrin? See how small it is? No linker. So it bundles the actin very close together into very tight contractile bundles, parallel bundles. And you can't fit anything in between there. You can't fit a huge motor protein. And so you're not, if you say that this is, that you would find fibrin in contractile actin, you would be wrong because you cannot actually even fit myosin 2 in here. Here there's a lot of space, so this is alpha-actinin, it has a larger linker. There's a larger space where you can actually have myosin 2 fitting in there. Looser packing by alpha-actinin allows myosin to enter, which makes these bundles contractile. Okay? Make sense? Yes?
Does that also happen with the ones that create webs, or is it just for the parallel?
Well, think of it in this sense. If you have a web, and then you tell me that fibrin is linking it together, well, then it's not a web.
Right.
And so if you see fibrin, you're not going to say this is a web, right? But if you see, filament, that's your big web one, then you can say that. So another example, out of control today. Villan is another cross-linking protein, and it's specifically, so it also has like 0 linker. So it tightly packs the actin in parallel bundles, and it's associated with formation of microvilli. Actually, if you overexpress villan in cells, those cells will form more microvilli because it's going to form these structures specifically. Okay? So filament has this kind of bent linker, right? These are kind of straight. This is bent. And so these are the ones that are used in forming these three-dimensional actin gels. And so that's kind of how it looks when it's bound to actin. So you can see how it stabilizes this gel. The red is the actin, the green is the filament. So this is required for motility. Questions? So filament is required for motility. So some cancer cells in malignant melanoma lose expression of filament. So here is a cancer cell that lost expression of filament. And here's a cancer cell that has filament. Do they look the same or different? So let's think a minute about filament and what it does, how the cells look. Would you say that this is good for cancer or bad for cancer? It's great for cancer. Why do you think it's great?
Because if they have with more filament, they can travel.
But, so with more filament, so if you have overexpression of filament, you can travel more. Absolutely. What if you lose expression?
Oh, I'm sorry. Then no, you can't travel.
Then you can't travel. So do you think, so is are traveling cells good, is that a good prognosis or bad prognosis? Bad, right? Metastasis. For metastatic melanoma, you need lots of motility, right? No motility, that's actually a good thing. So that's a less invasive form of melanoma would lose filament, and they can screen for this. So when you go in and you're diagnosed with cancer, they'll screen you for all sorts of different things that will help them to identify exactly what kind of cancer you have and how aggressive it is. So this is actually good for the patient, right? Any questions about this? So if this was on an exam, the way you would approach it is you would say to yourself, what is filament? Then you would remember it's a cross-linking protein associated with forming gels. It's associated with movement. And then you would ask yourself, okay, cell that can't move versus a cell that can move, which is going to be a worse cancer cell in terms of causing disease. I probably wouldn't ask you specifically about cancer because it's not a cancer class, but that would be how you would approach it. So that's how you bundle and connect actin filaments together, and also we spoke also about microtubules. So as you're going in your chart, right, we did all these different proteins, these accessory proteins. You can also make another line for proteins involved in organization or bundling of cytoskeletal proteins. And there we've got intermediate filaments we spoke about, microtubules we spoke about, and actin I spoke about. And so you can have a whole kind of a row of information there. So actin cytoskeletal structures need to attach to the plasma membrane. Right? Remember, actin is affecting the shape of the plasma membrane, so it makes sense it would have to attach, right? If actin's just floating in the cytoplasm, it's not going to have any effect on the plasma membrane. Remember we did the paper last week with E. coli? Who remembers last week? Okay. I don't. I don't even remember yesterday. No, just kidding. What'd you say? What? Breakfast? I got breakfast. No, seriously. We talked about EPEG. We talked about how it injects TIR, which is this receptor, and it gets phosphorylated and recruits ARP and WASP and makes the pedestal, right? Remember? So it's kind of recruiting these actin filaments to the plasma membrane and pushing the plasma membrane up. So try to kind of connect that paper with what we're talking about. Right, so you need to attach to the membrane, the plasma membrane in some way, or the cell cortex in some way. So these ERM family of proteins. Stands for Ezrin, Radixin, and mucin. And so this is a family of proteins that bind to the sides of actin filaments and the cytoplasmic tail of transmembrane glycoproteins. This is a good way to attach to the plasma membrane, right? If you attach to some sort of receptor or some transmembrane protein, then that's a good way. So the way it would work is you might have your ERM protein in some kind of folded inactive conformation. Then there would be some sort of signaling, right? Because remember, things just don't randomly happen. So maybe phosphorylation or PIP2 binding, some sort of signaling, and that would change the conformation of this protein so that it's available for interaction with the glycoprotein and then interaction with the actin filament, right? So it has a membrane binding domain and it has an actin binding domain. And so this is one way that you have that signaling can affect recruitment of actin filaments to the plasma membrane. And so these proteins, these ERM proteins, are targets of several different RTKs, right? What did we learn about RTKs? What are RTKs? Right, so they're receptor targeting kinases. So they're targets of RTKs. So that means RTK, so how does that work in your mind? If I say they're targets of RTKs, what does that mean to you? So ERM proteins have tyrosines that get phosphorylated by RTKs. Randomly or is it regulated? It's regulated, thank you. Right, so there's some sort of signaling event that activates an RTK, and then that RTA can phosphorylate ERM proteins and recruit actin to the plasma membrane. Remember we talked in the last exam about effector proteins and signaling and all this stuff, and I said, we talk about gene expression a lot, but often also the cell shape is being affected. And so here's an example of how signaling is going to affect, for example, cell shape and motility. Yes? No, RTKs are kinases, right? So RTKs are doing the phosphorylating of the ERM. Yeah. And that regulates activity. Any other questions? Hopefully at this point of the semester you're starting to be like, oh my god, it's all related, it's all one big thing. Or you're going, oh god, it's all one big thing. Really depends on how you're feeling about life right now. Okay. Should we talk about motor proteins? Why not? So there are a lot of movies that I have here because I feel like motor proteins are best spoken about with movies. And so the reason I was taking forever before is I was trying to get the movies up on the computer so that my computer doesn't freak out. And I think we're good. Okay, so motor proteins move along cytosol filaments, right? I keep talking about, it's like their tracks. There's these tracks. I think they're walking along them, right? And so there's certain things that you can kind of just always think about with motor proteins. They require ATP. There's always going to be ATP binding, ATP hydrolysis associated with the movement. There are many different kinds of motor proteins, and we'll talk about 3 different classes of motor proteins. Some move along actin, and some move along microtubules. They're specific to the track they walk on. Some carry cargo to different locations in the cell, and some cause cytoskeletal filaments to slide against each other, such as in muscle contraction, ciliary beating, and cell division. So here's your general ideas of motor proteins. So this picture is showing vesicles traveling along this cellular highway. So here's your microtubule, And only dynein and kinesin walk on microtubule. Myosin does not. Am I right? You guys believe me? And so it's showing you've got your motor domain or your head domain that is binding to the track. And generally. And that motor domain, since it's the part that's moving, that's the part that's binding to and hydrolyzing ATP. And then these different proteins, these motor proteins, they cycle between nucleotide binding, nucleotide hydrolysis, some kind of conformational change that's driven by the ATP binding state, filament release, some kind of conformational change. So it's kind of this like bind ATP, hydrolyze, move, bind. So it's just this walking movement. The motor domain determines the identity of the track. Yes?
So both line motor domain or head domain associated with
Yes, yes. Sometimes you'll hear motor domain and sometimes you'll hear head domain. It's the same thing. It's the part that's binding to the track. Yep. So the motor domain determines the identity of the track in the direction of movement. Some motor proteins walk to the plus end, some walk to the minus end. And the tail, so that's this part here, determines the identity of the cargo. So some will bind to vesicles, some will bind to other microtubules, some will bind to the cell cortex, and so we'll kind of go through examples of what these things look like. Do you guys remember the beginning of the semester when I showed you that movie? It showed very nicely a motor protein carrying a vesicle. So if you're feeling uninspired by this information, go back and watch that movie, and hopefully it'll be exciting. Yes.
Which part was the motor
The part that walks along the track.
and the tail
The tail, the motor domain or head domain binds to the track, so either the microtubule or the actin, and the head domain, not the head domain, the tail determines the identity of the cargo meaning the thing that's being moved. Yes?
But can you do 13, not the
Yeah, it's not a motor protein. I know, don't you hate when I do that? Kinesin 13 is not a motor protein. Well, you know what we should do? We should look up, let's do it just for fun. We'll look up the actual structure, domain, organization of kinesin 13. So the question I was getting here was, I'm predicting your question since I didn't allow you to finish asking it, but you're thinking, kinesin, motor protein. Wait, kinesin 13 is a catastrophe factor. Is that a motor protein? Is that what you're thinking? Well, it's not, right? Well, why is it called kinesin? Right? And so I wonder what the history is of kinesin 13. If it has some kind of structure that's similar, and it just maybe has no ATP binding domain, or maybe it does have some similarities in function to a motor protein, but functions differently. I have no idea. But kinesin 13 actually has a structure, at least in the textbook, that's kind of like this. And so, that's the crystal structure. No, it looks like a wishbone. And so it does make me think, based on the fact that kinesins have that kind of long tail, and then the two head domains, that maybe it has a similar structure, and so maybe originally when it was identified, it had some similarities, it was named that way. But we should look it up for fun. Let's make it a stump the teacher, because it's a very open-ended question. But really we want to know is, why is kinesin 13 named kinesin 13 if it is not a motor protein? And so that's not very Googleable. That's a thinking question. That requires you to read a little bit about kinesin 13. Look at the domain organization of kinesin 13. Maybe learn about why they named it what it is. I'll try to put up a thing on Blackboard to submit that to. It's been a while. Huh? People are just, it's that time of the semester where everyone wants more points. That's why I'm giving you Stump the Teacher. I want it's not even really a Stump the Teacher. It's just like a fun thought question. Okay, let's keep moving. So let's now talk about the different motor proteins. So there's the myosin family. It's a myosin superfamily of motor proteins. These are our actin-based motor proteins. These motor proteins only walk along actin. So if there's something on the exam where you have to remember what's what, remember myosin. You should always remember actin and myosin because they're kind of like two peas in a pod, right? But just remember that. This is the basic structure. You've got your motor, your head domains, and these light chains that are important for understanding the movement. And this neck and hinge region, also important for the movement. Then you've got this very long coil domain of two alpha helices. And then that coil actually will bundle with other myosin, or myosin, the contractile myosin that you're familiar with muscle contraction, myosin 2. And then the C-terminus is where your tail would be if you were going to be interacting with some other cargo. Right, so myosin 2 forms the bipolar thick filament that you've learned about with muscle contraction. And so it's a whole bunch of myosins. It's not one. It's a whole bunch of them that are organized bipolar. So some are facing this way with their heads this way, and some are facing this way. And those long coil domains are wrapped around each other. And that's how the bipolar thick filament forms. So these are hundreds of myosin heads, and they're oriented in the opposite directions. So they walk to the opposite direction. And they use ATP hydrolysis to walk to the plus end of the actin filament, and they slide pairs of actin filaments past each other. So that's contraction, right? So that's a close-up of how it looks. So here's your myosin tail. So this is one myosin 2. And you've got your heads here and your tail here. And so they're all kind of organized together with this coil domains bundled together. Did you guys ever see it like this before? Is that how you learned about it? Or did you learn about it at a much larger scale? Or who's never heard of myosin? Okay, so you can take ATP and actin and add them to myosin heads that are stuck to a glass slide. And those actin filaments will glide along the surface because all you need are those heads and ATP, and those heads are going to kind of start going like this. And so they want to bind to that actin. And so since they're moving like this, they're going to slide that actin around on the glass slide. And so here what they did is they were showing the movement of two different actin filaments. And so if you follow it over time, you can see it's here, then it's here, then it's here, and so on. The ATP is required for myosin function. I didn't even do anything. Look at that. Okay, so what I'm going to do is I'm going to stop the recording and save it now.